Good afternoon. My name is Caitlin Oots, and this summer I've been working with Dr. Triplett to uncover the life on leaves, more specifically, the interplay between bacteria and protists. First off, I wanted to introduce the phylosphere. It is the above-ground portion of a plant that serves as a habitat for microorganisms. This is important for the health of the plant because it protects them from environmental stress, pathogens, and helps regulate plant functions. The phylosphere plays a crucial role in the quality and yield of agricultural products. The phylosphere is host to many microorganisms, including a species of bacteria that I used in my experiments called Pantoea agglomerans. This bacteria is highly prevalent in the environment and is most commonly found in soils and on plants. Pantoea exhibits impressive nitrogen fixing capabilities, and because of this, it can enhance plant growth. Pantoea are very unique because they can form multicellular structures called symplasmata. These structures can contain hundreds of bacterial cells that are bound by a membrane. It is possible that these structures help safeguard the bacteria from UV radiation and desiccation stress. Additionally, the size of symplasmata may provide protection against predation. Last year, an undergrad showed that root protist induced symplasmata production, and this summer I have set up 5 experiments to see if leaf protist can do the same. For each experiment, I used 2 different protists from the same genus, Colpoda, one found in the roots and another found on leaves. I also used the symplasmata-forming bacteria, Pantoea. In all 5 of my experiments, I set up a 4x6 well plate with various mixtures of the bacteria and protists. I then performed serial dilutions and pipetted the different mixtures onto LB agar plates to obtain bacterial counts. I also took pictures of each of the wells from the 4x6 plates to count the number of symplasmata and protists per milliliter to determine if the leaf protist induced symplasmata. I found that leaf protists caused a decline in the number of symplasmata compared to the root protists. As observed in the graph, the symplasmata counts rapidly decreased with the leaf protists, while a low population remained stable with the root protists. These pictures clearly show that the presence of leaf protists, existing symplasmata disappeared. This graph shows the average root protists in each well, and it was observed that the root protist grew better on its own and had reduced growth when mixed with Pantoea. However, the leaf protist population was 10 times higher when mixed with Pantoea compared to when it was alone. This experiment demonstrated that Pantoea bacteria enhanced the growth of leaf protists, but suppressed the growth of root protists. For one of my experiments, I used a constitutive red reporter and mixed it with a GFP-labeled symplasmata reporter. This setup caused all of the single bacterial cells to glow red and the symplasmata to glow green. The graph indicates that the root protist emitted more green fluorescence than the leaf protists. This is because more symplasmata formed with the root protists, possibly to protect themselves from predation, while the leaf protists inhibited symplasmata growth and consumed more bacteria. Therefore, it had less green admission. I recorded this video to show the leaf protist glowing from ingesting the bacteria and how none of the symplasmata were able to be consumed by the protists. The key insights from my experiments were that leaf protists cause symplasmata to decline, but root protists do not. Pantoea suppressed the growth of root protists, but enhanced the growth of leaf protists, and root protists induce symplasmata genes, but leaf protists suppress them. From these conclusions, it appears that symplasmata formation in response to predators is specific to the root protists, while leaf protists do not induce symplasmata and are well adapted to Pantoea. This is interesting because it was previously believed that leaf protists survive due to their adaptations to harsh environments. However, it may be due to a mutualistic relationship with leaf bacteria. I just wanted to give a shout out to my mentor, Dr. Triplett, and the team I worked with here at the lab. Thank you guys so much for listening, and I hope you guys have a wonderful day.